Thank you for the invitation. A lot of fancy word for the presentation. Actually, I have no idea why I'm here because I don't know anything about conversion rate, for instance. This is a word that I don't know. But I think I'm here because I will talk about crap. Because my talk is named How DNA Sequencing Can Change Our Society, but I could have named it How Crap Sequencing is Changing Our Society. So I will try to be not too technical. I will start from the beginning, explaining what is DNA, how we sequence it, why, and what are some interesting applications, including the gut microbiome assessment, which is a big thing in toxicology and in medicine nowadays, because we learn that bacteria which are in our gut have a major effect on our health. So the human genome, to give a start, I can say that it's like a book. I actually like to compare it to the book of life because it contains the instruction to make life. And it's like a book because it's written with an alphabet of 4 letters, A, T, G, C. And these letters make the human genome. This human genome is even separated in different chapters, 46 chapters, the 46 chromosomes. And this human genome is gigantic. The size of the human genome is 3 gigabytes, 3 gigabases, 3 billions of these letters. And if you print the human genome, it's as big as 600 of the 7 Harry Potter books. And this human genome can tell us a lot about who you are and your health. It's actually also, this book of life is actually also made of sentences. These sentences are the genes. They are giving a function, an instruction to make life. And the sequencing of the human genome has been one of the greatest challenges in scientific history. Last decades have been spent to sequence the human genome. The cost was astronomical. It was $1 billion to sequence the first human genome. It took 30 years. Nowadays, we can sequence a human genome for $1,000. We even have now portable real-time device for DNA sequencing, very small machines. 10 years ago, the DNA sequencers were like these big machines, as big as a fridge, and now we can have very small device that can fit in a pocket, so you can sequence DNA everywhere. I actually have one of these here. I can show you how it works later. I don't have 3 hands. So this device that I took, it's one of the latest generation of DNA sequencer. It's very small. You just have to plug it to a laptop. And what you do is that you just put your DNA sample inside this device, and there is a membrane which contains a lot of holes like that, and the DNA strand will go through these holes. And when there is a change in electric current, in this case, you can see if it's an A, a T, a G, or a C. This is called the nanopore technology, and this is one of the most promising advances for DNA sequencing. DNA sequencing is quite complicated, and generally we don't sequence a human genome at once. What we do is that we take the DNA from the genome, we break it in very small pieces that are generally 100 letters long, like these ones, and we sequence, we look at the DNA sequence composition for around a million of this DNA string. We generate 100 gigabytes of data. Then, to understand this data and to analyze it, it takes a lot of computer power because you have to make a gigantic puzzle with these million DNA strings to put them together and to rebuild a human genome. If I have to say for one human genome, you need maybe 1 day on a high-throughput computing system. On my laptop, it would be 1 week. And this generates a lot of data because if you want to sequence the human genome of the whole British population, it will be a lot of data. It is estimated that approximately 40 million terabytes of storage will be needed by 2025 just for the human genome. And ironically, one of the most promising ways to store data in the future is DNA itself. Because we can think about replacing DNA, hard drives with DNA. It will be slower to read the data and write the data, but it's a very promising way for long-term storage. DNA can be actually retrieved from samples of prehistoric animals which lived 700,000 years ago. So you can see that's a very long storage. It's still early days, but it's a very promising technology. What you can see here is the first video which was encoded in DNA. It was put in a bacteria. This bacteria was grown. It could be kept forever. And then this original image was reconstituted from the bacteria. And you can see that for a first try, it's quite convincing. It's a 5-frame GIF. It's about 30 pixels long and 20 pixels large, 2.6 kilobytes. And this is quite a milestone for storage of DNA, storage of information into DNA. There's actually a news which was released quite recently about this song of Deep Purple, Smoke on the Water, which was also encoded in DNA. This gives me a bit of opportunity to explain you how it works in this case. You can take the 4 bases of the DNA sequence and assemble them in triplets for each character, space, and punctuation. And for instance, the word smoke will become GAC for an S, and so on. It's still early days for this technology, but the cost of this technology is decreasing quite quickly. For storing 12 megabytes of data in DNA, which was the song Smoke of the Water, it costed around $100,000. So that's a lot of money, but when you look at this curve, the price per DNA letter of synthetic genes over time is decreasing a lot. It's decreasing by 10 every 4 years. So we can imagine that in 10, 20 years, we will be able to use DNA for storing information. I've seen an article, it was yesterday, they were saying that it's a 30 billion market, something like that. But actually, the most useful way of DNA sequencing is probably to save life by what we call genomic medicine. One of the most telling examples is to cure cancer, because a cancer is an abnormal proliferation of tissue. And this tissue will have different DNA from your normal tissue. And this abnormal DNA can be sequenced, identified, and you can develop strategies for personalized assay for chemotherapy. And this is something which is already done classically for some cancer, and this has a very high success rate. Now I want to talk about consumer genetic testing, because you can find a lot of articles talking about the success of genomic medicines, but it's always a bit more difficult to be critical. What I call consumer genetic testing, it's these companies which are promising you to reveal that you can, to tell you if you can eat gluten, if your ancestors are Viking, if you have a high IQ, this type of thing. You can see them actually making advertising everywhere in the underground. One of the most common examples is the company 23andMe. I quite like this cartoon because it's not that wrong, because last year there was a report of some scientists who sent some DNA to one of these companies. They sent their dog's DNA, and the company couldn't say if it's a dog or human. And these guys received a report saying that this individual has very particular skills, and that it could be very excellent for gymnastics and boxing. So we should be careful with this type of promises and this type of test. It's quite good in a way because it puts DNA sequencing and interpretation at the consumer level. And it could be interesting to know some things about ancestry, for instance, but it could be very dangerous and if it's applied for clinical interpretations. These companies, they will tell you if you have more risk to develop a cancer, to have Alzheimer, or to have Parkinson, any disease, but this is not that easy to say that you can develop a disease based on your genome. There is always an error, and these companies play on this, but this is not actually that smart. Last year, a clinical lab checked some results from one of these companies, and they found that 40% of their clinical interpretation about genes associated to cancer were false positive. This is actually worrying because some of these genes associated to cancer can be very, very powerful and can give cancer to sometimes very high rates. This is the case of Angelina Jolie. She had an ablation of breast and ovaries because she was carrying a gene BRCA1, which gives individual a probability of developing cancer of 80%. So DNA sequencing could be useful, but you have to be careful about interpretation and always be cautious. I like this quote from a famous British geneticist saying that these companies are asking people to pay for something which is actually at best trivial and at worst astrology. But we are actually not just the sum of our genes. There is a strong impact of our environment. And now I will talk about the gut microbiome, because all the bacteria which are in our environment are affecting us a lot. Take twins, for instance. They have the same genome, but they will develop disease, and they will not develop disease at the same time. They will have each their path in life. This picture is a piece of art, but it's actually quite telling because this artist took the bacteria which was on her skin and just made them grow on this structure to show people that we are actually swimming in bacteria. We have bacteria everywhere. And it's not a problem because most of these bacteria are beneficial. We are always in the mindset that bacteria are harmful and that hygiene is very important. We should be careful, but in many cases, these bacteria will protect us. We have actually quite a lot of them in our body, and we have most of them in the gut microbiome. We have 10 to 100 trillion microbes in the gut. It's around 1 kilo. In my daily job, this is what I'm analyzing. I can say that I like analyzing the data, but I don't want to be the person collecting the samples. And DNA sequencing was quite a revolution to study the gut microbiome because before the advent of DNA sequencing, it was impossible to analyze these bacteria because they cannot be cultured. So you have to extract their DNA, sequence their DNA to identify them. The human gut microbiome has profound effect on human health. It can influence virtually anything. Your mood, if you are hungry or not, the way you accumulate fat, the way you digest food. It's actually linked to many diseases like diabetes, obesity, Parkinson, autism. In some cases, autistic kids, you give them a probiotic and they will improve, showing that it's a very important aspect of the disease. I like this example because this is a study which was done in London by one of my collaborators, and they were working on twins. They took twins, one which is obese and one which is lean. And they were wondering why they are that different even if they have the same genome. And what they did is to take the microbiome from one twin which is obese. They put it in a mouse. They did the same from the lean twin, and they found out that the mouse which was exposed to the microbiome of the obese person got fat. This is actually a new technique which is not like a diet, but for persons which are in morbid obesity and at risk, fecal transplant, which is a bit disgusting if we talk about that. Sometimes we call that capsule, because you can have these small capsules that you can eat, and this is transmitting the microbiome of someone who is considered as healthy. I also like to take examples from the wildlife because this is very interesting to see that it's also very important for animals. If koalas didn't have their gut microbiome, they couldn't survive because young koalas, they have to consume the feces of their parents to obtain the bacteria allowing them to digest the food. And this is the basis of what I told you, the fecal transplants, the actual capsule, which are very successful to cure obesity and also bacterial gut infection. I kept the picture of the koala because I looked on Google at the fecal transplant and you don't want to do that. In humans, this is the same. Our digestion is influenced by the gut microbiome. Japanese individuals, if they can digest the algae nori more than any other people, it's because of their gut microbiome. It's because they have one bacteria which colonized their gut and which allows them to digest this type of algae. Gut microbiome, it affects also the efficiency of common medication. Some people will not respond to chemotherapy because of their gut microbiome. Because the gut bacteria, they will munch on the drugs, and so if they are there, it's becoming very inefficient. Sometimes they will make the drug even more toxic. So this is actually now quite a common practice to check for the gut microbiome composition when you give a drug to someone, so you can make sure that this person is compatible. I found this article yesterday when I was preparing the talk that it's not just the gut microbiome, the oral microbiome is also very important because in this case, these people, they found out that the bacteria which are in your mouth, they can be distinct and predictive for people developing colorectal cancer. So very interesting because now you don't have to do a coloscopy, you can just do a swab of your mouth and maybe you will have an indication if you have a problem or not. My work is to study the effect of pesticides on the gut microbiome. I haven't talked much about that, and I don't know if I have much time to talk about it. So I will be quick about that. And so I'm looking at the effect of pesticides on the gut microbiome because pesticides are everywhere in our food. There's virtually no foodstuff which doesn't have pesticides, even organic food. And these pesticides that are put on food, on crops, they end up in your body, and everyone will have pesticide in their urine if I measure them today. Especially one of the most useful for farmers, which is called glyphosate. It's in the news quite often these days because it has been linked to cancer. And one of my main research topics is to ask if glyphosate can have an effect on the gut microbiome. Because these pesticides, they are made to be non-toxic to human cells, but they act on plants and bacteria, fungi in some cases, so they can actually disturb your gut microbiome and cause some disease. But this is something which has never been evaluated because this is new to science. And I will stop here before I go too technical. Thank you.